Alright guys, we've been talking about the cell cycle, mostly focusing on interphase, and now I want to move on to the mitotic phase, which includes mitosis right here. So we were talking about all this up here, now we're going to look at the blue, mostly mitosis. We do need to remember the stages of mitosis, and remember that mitosis is a copy machine. So you're going to make an exact copy of a cell. But really, in the mitosis phase, we're not making an exact copy of the cell, we're making an exact copy of everything in the nucleus. It's nuclear division. So those stages that you need to remember are prophase, metaphase, anaphase, and telophase. And I'm going to give you ways to remember what happens in each of these phases. To know the order of them, remember PMAT, P-M-A-T, PMAT, prophase, metaphase, anaphase, telophase. And I'll explain these a little bit later, but here's some hints for you. In metaphase, you can think of middle, meta, middle. Anaphase, you can think of Anna being a mean girl, and I'll show you that in a little bit as well. But you will need to know the phases of mitosis and the fact that mitosis is simply a copy machine making exact copies of the nucleus. So it's nuclear division. Now the whole cell division is going to be in cytokinesis, which we're also going to talk about. Okay, the first phase is prophase. Prophase is where nuclear division is about to occur. So what has to happen is chromatin is going to be condensed. Remember, chromatin is your DNA that looks like spaghetti in a bowl. And that's when it's being used to make proteins. Now, before nuclear division, it needs to be organized. So wrapped up kind of like your Christmas lights, it's going to be organized into these chromosomes. And then they're also organized by their copies. So remember, chromosomes were copied in interphase. So those chromosomes that were copied in interphase are connected by where they're holding hands right here. And those are called centromeres, those points where they're holding hands. And each copy of the chromosome is a sister chromatid. They're holding hands. Okay, so that's what the DNA is going to look like right now. Now in prophase, the nucleolus, this little part in the center of the nucleus that we talked about in the last chapter, it's going to start to disappear, and it is going to disappear. The nuclear envelope, so the membrane around the nucleus, it's going to fragment and eventually dissolve. And these spindles are going to start forming. Spindles are those Spider-Man webs that we've talked about before. The Spider-Man webs that come out of the centrioles, which are in the centrosome. The centrosome is the organelle. The centrioles contain the spiderweb fluid, and the spindles are the spider webs. So that spindle is going to form. It's going to hold the chromosomes in place where they need to be. And then those two centrosomes are going to move away. So what they've done here is broken it into from early phase to late prophase. So early prophase, late prophase. We're just going to focus on prophase in general. They split it up because so much happens during prophase, but I won't ask you which parts are early prophase and which parts are late prophase. Just know that all of these events happen during prophase. Okay, so our nucleus is gone. We have our centrosomes, and we have spindle fibers here that are attached to all these chromosomes that are lined up in X's in the middle. And those X's are composed of sister chromatids, so copies of the same chromosome that are holding hands, okay, at the centromere. And then we go into metaphase. During metaphase, that's where the chromosomes are going to line up down the middle. That's where middle comes from. And they're all going to be connected by these spindle fibers and held in place right in the middle. And remember, this is also where a gateway checkpoint is, to make sure that all of those chromosomes are where they're supposed to be and lined up correctly on the spindle fibers. Otherwise, we may have to abort if things didn't go as they were supposed to. Yeah, so that's where we're talking about that. It's called the M checkpoint. Now, I also wanted to stop here to remind you that we have some confusing vocabulary words that you need to make sure you can keep separate. So remember, these different words are centriole. Centriole is going to be these structures here that hold the Spider-Man fluid in there. Remember the spindles? They're going to shoot out the spindles. The centrosome is the organelle that contains the centriole. The centromere is going to be where the sister chromatids are holding hands right here. A chromosome is going to be the condensed DNA, as you can see. Sister chromatids, each half of this X is going to be a sister chromatid. They're exact copies holding hands, like kids holding hands. When these come apart, which we don't have yet, eventually these sister chromatids will break apart. That's going to be our next slide. Then they're considered daughter chromosomes. And remember, chromatin was the loose DNA that looked like spaghetti in a bowl that can actually be used to make proteins. Okay, so throughout this whole thing, make sure you keep reminding yourself of what these vocab words are and how to keep them separate. All right, the next stage of mitosis is anaphase, and this is where that mean girl Anna is going to come in and break apart the sisters, if you want to think of it that way. This is where the sisters let go of their hands and come apart. So they're no longer sister chromatids, now they're daughter chromosomes. They've been broken apart. That is what happens in anaphase, because those centromeres that were holding them together dissolves, releasing sister chromatids, and the sister chromatids become daughter chromosomes. The spindles will actually pull to opposite poles along the fibers so that they will move apart. Okay. And then we come to our last phase, which is the telophase. This is where the spindle is going to disappear, so we won't have the Spider-Man webs holding on to the chromosomes anymore. And we're going to have a new nuclear membrane forming. So there's going to be two nuclear membranes now. All of that stuff that happened in prophase where the membrane disappeared, the spindle fibers forms, we're doing this in reverse now. Spindle fibers go away and the nuclear membranes are going to form around two nuclei now. One cell, two nuclei. That's how you know you're in telophase. Let's see. And each daughter nuclei is going to receive one chromosome of each type. I'm going to make sure that all chromosomes are represented in each of those nuclei. And then the chromosomes are going to unspool to become diffused chromatin again. So the pieces of DNA that they are going to be required for that cell, what types of protein are required for that cell, those are going to unravel so that they can be made again. Whereas the ones that are not required for that cell will stay packed up in their boxes. Okay, then we go on to our next step. This is not part of mitosis, but it is part of the mitotic phase. I know that can be confusing as well. Mitosis was the nuclear division, where cytokinesis is going to be the division of the whole cell, or the cytoplasm. That's actually what cytokinesis means. It means division of cytoplasm. So we're going to have the mother cell's cytoplasm is going to be divided equally into each daughter nuclei, or each daughter cell actually. And then we're going to have this cleavage furrow form in the cell membrane. And this happens with the contractile ring, which is kind of still the Spider-Man idea. And it's going to pull apart like a drawstring bag. Like pulling on a drawstring, eventually those two pieces of the cell right here are going to pinch off and become two daughter cells. Okay, we've been looking at all of these drawings, representations of the different phases of mitosis. Let's see what it actually looks like when you're looking at actual cells under the microscope. And I'm going to have some of these available for you to use for your lab this week. I'll explain that in detail in emails and so forth for you guys in a minute. But what I want you to see is all of these up here in blue, these are animal cells, and all of these down here in green are plant cells. Oh, I forgot to mention that this right here would be an animal cell, how the cleavage furrow would form to make two different daughter cells. In plant cells, because there's a cell wall in plant cells, they cannot do a cleavage furrow. So a whole new cell wall or a cell plate has to form. So we're going to look at that when we look at these pictures. I'm going to go through this really quickly because I know you can read these bubbles and look at this at home, and I only have 15 minutes of video. So they've broken it down into early prophase, prophase, prometaphase. So prometaphase is going to be just right before metaphase, the early stage of metaphase. Then you have metaphase where they're actually lined up down the middle. Anaphase and telophase, and then there is kind of a continuum, so it's hard to tell when you're looking at actual cells which phase they're in sometimes, and know that sometimes they're in between phases. So use your best judgment when you're asked which phase a cell is in, and we can talk about that in more detail in a minute. I do want to show you, this is cytokinesis in an animal cell. Really cool pictures. You can see the actual image of animal cells next to a drawing. So we have the cleavage furrow forming here, and then the contractile ring with these spindles actually pull apart really far, and then they will close off and form two different daughter cells. That's an animal cell for cytokinesis. But in a plant cell, remember, we can't have a cleavage furrow in animal cells because they have a cell wall. So there's a cell plate that has to form. And that cell plate is going to be formed by these vacuoles that bring in materials and build like a new wall right between those two new daughter cells. So it's slightly different because of that cell wall in plant cells. Now, when does this happen? We've talked about mitosis, but why do cells need to undergo mitosis? Well, the function of mitosis in general is just to permit growth and repair. That's it. So within your body, mitosis is necessary when a fertilized egg becomes an embryo, or an embryo becomes a fetus, or after birth when a child becomes an adult. So all of this growth that's happening, all of this growth, it's not just because cells get bigger, it's because there's more cells. You need more cells. So mitosis is going to make exact copies of cells and make more and more and more and more. You can also see this in trees. Trees increase their girth or their width each growing season. So if you've ever counted the rings of a tree to see how old they are, that is because more cells have been made in certain years when you can even tell if it was a good growing season or not. If it's a really thick ring, then you know a whole bunch of cells were laid down in that year or that season. So it tells you a lot about plants as well when you can look at how much mitosis occurred, how much growth happened. Now another function of mitosis is going to be, I guess I did put that here. Here, we'll talk about that in a minute. There is another purpose of mitosis, and that's going to be asexual reproduction, but that only happens in single-celled organisms. So reproduction itself is not mitosis. We're going to talk about that in a minute. But asexual reproduction, that means reproduction without sex, that is going to happen in single-celled organisms. And that's called binary fission also. So let's look at prokaryotic cells because they are single cells and they're completely different. How are prokaryotes different from eukaryotes? Remember, prokaryotes are very, very simple. They do not have organelles. They don't have a nucleus. They do have DNA, however. So they have a section of DNA, and it's actually a ring of DNA, so it's in a circle here. And the area in the cell where the DNA is, that's called a nucleoid region. There's no nuclear membrane or anything like that. But the DNA is still replicated. And then the cell actually splits into two during binary fission. When it splits into binary fission, you have a whole nother cell. And it's a whole nother organism because they're single-celled organisms. So because it's a whole nother organism, those two daughter cells are actually produced through asexual reproduction. We can't do that. We can't just split ourselves in half and be a whole new organism. So we do not do asexual reproduction. But mitosis of a prokaryotic cell does lead to asexual reproduction. I only have 30 seconds, so I'm going to have to end it there. But I really, really miss you guys. And if you have any questions, make sure that you email me or call me. And this is going to be a cell that you can, a chart that you can look at to see what I was talking about with the functions. Eukaryotes, if they're single-celled, can also do asexual reproduction. But plants, animals, fungi, we cannot. We only do development, growth, and repair for mitosis because we cannot do asexual reproduction. We cannot split in half and have a whole nother version of us. Also, for the, I got to end it there. Bye, guys.